everyone, my name is Melissa Patino-Martinez and I'm a rising senior at Dickinson College, and today I'm going to take you through sort of what my day today and summer in general, I guess, looked like at the Chicago Botanic Gardens. Here are some clips of my commute in the morning. So this summer I worked with my amazing, extraordinary mentor, Nora Gavin Smith, and we were working with Impatiens, which is a plant genus that is all over the world, but specifically we focused on those endemic to the Eastern Rocky Mountains in Tanzania. You'll notice them in the red markers. This is Impatiens angleri, and this is Impatiens kylii. And they're super cool because Impatiens angleri is mostly butterfly pollinated, while Impatiens kylii is mostly bird pollinated. And so here we are in the lab. Here I am holding a multi-channel, and we're zooming into a 96-hole plate and a little trough, and these are kind of what my setup looked like. Now we'll be taking our 96-hole plate from the centrifuge. It is a machine that spins at super fast speeds and has to be balanced, and anything that you have in it will be sent to the bottom. And it's also really cool when you want to separate liquids from solids. So here I am just transferring my sample to the other side of my 96-hole plate, and then after some cleaning and a few other things, I am now checking for concentration. This is towards the end of something called library prep, and it basically is a protocol that cleans up DNA, sizes it for specific sizes. Our target size was 500 base pairs, and then with a bunch of cleanups and a barcoding and polymerase chain reaction, which just makes a lot of copies of it, you get copies of the target DNA, and then you can make libraries. Here I am taking about 2 microliters of the sample and putting them into these microtubes so that I can qubit them and check for concentration. The DNA sample is also added to about 198 microliters of a dye solution that binds to the DNA, which then fluoresces in the qubit machine, which tells us how much there is in our sample. Now we're taking a little quick break and walking through the gardens to one of my favorite lunch spots. About like a month and a half in, I was like, I should sit outside and eat lunch. And so here I am sitting eating lunch. And one of these days, when I was walking back with Julia, a little surprise guest appearance. And I have lunch with a few other of the REU interns. Very cool. Some great friends that I've made along the way. And so here's another little clip of our walk back to the Plant Science Center from having a tasty lunch with a great view. Now that we're back in the lab, I am pulling up my coding. It is very cool, honestly. I feel like I'm in the Matrix. Like, I thought I was super cool when I would be typing the things in. You'll notice that sometimes the screen flashes like a super pale yellow or something. That's me hitting the tab. It tells the command line to fills in the blanks for me. So now I'm pulling up a genome for one of the samples. It is super long, which is where the program to like clean up everything and like make it more human readable and user friendly comes in, and it was a lot of work, but it was super cool in the end. And I also wanted to show a picture of like when things don't go right, because sometimes things don't go right and that's okay, and we troubleshooted and it was great. And so here I am done with the day, walking out. I had tons of fun, and this was a great summer. Some fun things that I really liked were sonication, which is where you take a sample and put it in a little water bath in a machine, and then hit it with a specific frequency of sound waves so that it breaks down into the target sizes. Here is a clip of a little deer that I saw. But yeah, anyways, that concludes my summer Wombo Combo video. And I hope you liked it, and a huge thank you to Nora, the RU coordinators, my friends who also happen to be RU interns, their mentors, and just literally everybody else that made the days super, super great and worth it.